Video 12.3 is on a topic that we've talked about before, mutations. Mutations result from changes in the DNA. We've talked about mutations in our evolution unit as a mechanism for genetic variation, and we talked about mutations in our last unit when meiosis goes wrong with crossing over or as a result of non-disjunction errors during anaphase. In this video, we'll focus on one objective. This video will focus on how changes in DNA can result in changes in protein structure, or not. So let's start by refining our definition of a mutation. Yes, a mutation is a change in DNA sequence, but a mutation is a stable and heritable change in DNA. This means that the DNA change isn't corrected by a proofreading polymerase like DNA polymerase I, or the DNA change isn't detected by p53 and then corrected. As a result, the DNA change results in a change in RNA that results in a change in protein. Since unicellular organisms reproduce by dividing their whole cell, the mutation is passed on to all of their offspring. In multicellular organisms, the story is a bit different. If the mutation is in a somatic body cell, the mutation stays in the organism. But if the mutation is in a gamete, then the mutation will be passed on to the offspring. We've talked about some types of mutation before. In our genetic unit, we covered chromosome imbalances and macromutations. The new information for this video is about point mutations. The types of point mutations we'll cover include missense, nonsense, read-through, and frameshift mutations as a result of nucleotide insertions or deletions. Point mutations are considered small-scale mutations. Point mutations are caused by errors in DNA replication or other environmental factors. If you think about DNA replication, though, the errors would be created by DNA polymerase III and would go undetected and uncorrected by DNA polymerase I. Also, the errors would be undetected by p53. In contrast, macromutations and chromosome imbalances are large-scale mutations caused by errors in crossing over and errors during anaphase. So what exactly is a point mutation? Think of a point mutation as a change in one point on the DNA. In other words, just a single base change. What can possibly change about a base? Well, one base can be changed into another base, and that's called a substitution. An extra base can be added, called an insertion, or a base can be deleted. Most of these errors are caused by DNA polymerase when cells replicate their DNA during replication, but these errors can also be caused by environmental factors like chemicals, radiation, or viruses. Any factor that causes mutations is called mutagens. I want to consider each type of point mutation in a case-by-case way. First up are all of the types of mutations caused by substitution. Mutations can actually be harmless. A harmless mutation is called a silent mutation. As a result of the change in a base, there is still no change in amino acid. As you can see in this diagram, the normal codon sequence of GCC codes for the amino acid glycine, but a change of GCC to GGU still codes for glycine. So a silent mutation changes the DNA without changing the amino acid. But a substitution can also change the amino acid, and this would be called a missense mutation. In this example, GGC codes for glycine, but AGC switches the amino acid to serine. Similarly, a nonsense mutation changes an amino acid to something else, but a nonsense mutation switches an amino acid to a stop codon. So in this example, you can see that AAG codes for lysine, but a point mutation that switches the codon to UAG codes for a stop codon. The result is a polypeptide that's a lot shorter than what it's supposed to be. This is a premature stop codon. The opposite effect to this that's not pictured would be called a read-through mutation. A read-through mutation takes where there should normally be a stop codon, and it turns that stop codon into an amino acid instead. So the polypeptide isn't stopped, and instead the polypeptide turns into a protein that can be extra long. Besides substitution, there are other types of point mutations that insert or delete a nucleotide. Check out the example. Here you see UUU coding for phenylalanine in the wild type. A mutation deletes one of the U nucleotides, and so instead of phenylalanine, the result is leucine. But also, every downstream codon is changed. This is because inserting or deleting a nucleotide changes the reading frame. Here's what I mean. Read this sentence. Why did the red bat eat the fat rat? Reading that sentence, your brain separates the letters into 3-letter words. But what if there's a deletion, like deleting this D right here? Why did the rebate blah? Every word after this D deletion is messed up. The same is true with an insertion. Here, the normal sequence of AAG is a codon for lysine, but an insertion of a U nucleotide introduces a premature stop codon. Now that protein will really be messed up. An example of a mutation is in sickle cell anemia, where there's a mutation in the oxygen-carrying protein hemoglobin. And check out the mutation here. The CTT DNA sequence is mutated into CAT. This is a missense mutation that results in a change of the mRNA codon from GAA to GUA, and the protein has a valine amino acid instead of the normal glutamic acid. No big deal, right? It's just one amino acid. But it is a big deal. Glutamic acid has a hydrophilic R group and valine has a hydrophobic R group. What this means is that to be stable, hydrophobic amino acids of the mutant hemoglobins stick to each other. The mutant hemoglobins form really long chains that deform the red blood cells like this. These red blood cells are shaped like a sickle, one of those farm tools that looks kind of like a moon. And this is bad because the deformed red blood cells can get stuck in small capillaries, and that can lead to big problems with tissues getting oxygen and getting rid of waste. Another example of point mutations causing human disease is in cystic fibrosis. A common mutation in cystic fibrosis is that the triplet of CTT nucleotides is missing. As a result of this deletion, you can see that the protein sequence is affected. The phenylalanine at position 508 is deleted and is no longer there. Isoleucine, isoleucine, glycine, valine, isoleucine, isoleucine, phenylalanine, glycine, valine. So why is cystic fibrosis so bad? The mutation is in a chloride channel that's responsible for regulating the viscosity or thickness of mucus in places like your lungs. So without treatment, a person with cystic fibrosis will only live about 5 years. With treatment, however, the lifespan is extended to somebody living into their 30s or 40s, and maybe even longer with recent advances in medicine. From a physiology standpoint, here's what happens. Normally, this chloride channel moves chloride ions from high to low by facilitated diffusion. As a result, water follows the chloride ions by osmosis, and the increased water keeps mucus dilute and watery. Watery mucus is good because it can trap bacteria and pollutants that get into your lungs when you breathe, but still be coughed up to clear those pollutants out of a person's lungs. When the chloride channel is mutated, however, chloride ions don't move. So water doesn't follow by osmosis, and the resulting mucus is really thick and viscous. Now, bacteria still get trapped, but the mucus is so thick that it can't be easily coughed up. So the mucus creates this perfect environment for bacteria to grow, out of control and cause infections, and that's a bad thing. Cystic fibrosis is a disease that can be treated if you can treat those infections and keep those infections away. You have to clear out that mucus that traps the infecting bacteria and prevent the infections from taking hold. One way to clear out the mucus is by percussive therapy. In infants, you can tap on the patient's thoracic cavity to help loosen and clear that mucus. Older patients can use a percussive vest that helps tap their chest. Similarly, mucus thinners can make it easier to clear the mucus. Treatment with antibiotics can help prevent the bacteria from multiplying, kill the bacteria, and prevent infection. The best way to help cystic fibrosis, however, is actually with a lung transplant. The new lungs would be from a person that presumably doesn't have the cystic fibrosis mutation. Those lung cells and those new lungs would be fine and would make mucus that's the appropriate viscosity. So I've talked about sickle cell anemia and cystic fibrosis, and that makes mutations seem really bad. But remember, you need mutations to create variations that can be selected for or against by nature to create organisms that are more ideally suited for their environment. So are mutations really bad? That's something that I want you to think about. So let's recap what we've learned in this video. First, we refined our definition of mutations. Mutations are stable, heritable changes in DNA. The new type of mutations that I've covered in this video are point mutations that are caused by errors of DNA polymerase during DNA replication in the cell cycle, or by environmental factors like mutagens that change just one or a few DNA bases. Nucleotide substitutions can result in different mutations like silent mutations that really don't matter, nonsense mutations that change one amino acid to another, or missense mutations that change an amino acid into a stop codon. Similarly, point mutations that are insertions or deletions can create frame shift mutations that every place downstream of the mutation gets messed up by changing the amino acid, unless that insertion or deletion is in a group of 3. Even though mutations can be bad and cause diseases like sickle cell anemia and cystic fibrosis, mutations are a mechanism of genetic diversity that help drive evolution.